let me see if this advances. Okay, so here are my disclosures. So it's a little bit of a tongue-in-cheek title, but so really I want to talk to you about the tau protein therapeutic multiverse. And there's a lot to talk about. These are exciting times for those of us who want to develop treatments for neurodegenerative disease. And so I think to really effectively find treatments that target the tau protein, we have to look at everything, unfortunately. So there are different tauopathies. There are different tau genetic risk factors for each disease. There are different protein isoforms, different pathogenic mechanisms, different 3D aggregate structures of tau in different diseases, and there are different biomarker profiles and therapeutic approaches that likely we will need to take for the different tau-related diseases. We also have to think about where we're going to do this, and both on the molecular level, where in or around pathways that involve tau or on the tau protein itself. It's a complicated, alternatively spliced protein. Also, how can we do this in multi-center trials? And particularly, how can we improve the diversity of our clinical trials, both within the United States and North America, but also internationally, to involve people who have been historically not able to participate in disease-modifying clinical trials. And what we've learned, and these are exciting times for those who haven't really been involved in clinical development of dementia therapeutics, is that it's a very slow and expensive process to do clinical development and clinical trials. And so, my goal really is to think about how we can do this better or more precisely and more efficiently so that it doesn't take us as long to find effective tau therapies as it has for effective amyloid therapies. So this is from a old drug development review that sort of suggested that developing a new drug, possibly this could be for a CNS or non-CNS indication, might on average take 12 to 15 years, and it's sort of broken half, into the preclinical target identification and other aspects, and then into the clinical aspects of drug development or clinical trials. From my perspective as a neurologist, a practicing neurologist who take care of patients with neurodegenerative diseases, I really think about the preclinical work as hypothesis generating. You're coming up with drugs that you think might treat these diseases, and there are all these different questions you might ask. But ultimately, if we want to treat human disease, we have to test the hypothesis in human clinical trials. And, depending on the stage of development, these are some of the questions we might want to answer. Is the mechanism the same in humans as the model you used? Does the drug actually perturb that mechanism, and will that affect the biology? And ultimately, we care about whether it helps our patients. So if we look back, and this is my perspective, because my career, I haven't been doing this for 30 years yet, but I've been, early on in my career, I got involved with bapineuzumab, and after that, we ran many clinical trials with amyloid immunotherapies. And I would argue that, the actual preclinical portion of amyloid immunotherapy development didn't take very long. And really, the most time limiting and hardest part has been the clinical trial or clinical development component. And so, we're really focused on how can we do this better or faster so that it's not so difficult for this next generation of therapies. And specifically, I want to, talk about the idea of precision medicine or precision clinical trials. And again, I've learned a lot from Howard over the past number of years, because he's thought about this a lot too. But, if we think about recent successful clinical trial approaches, they're sort of, less precise approaches. And once in a while, for instance, if we think about sporadic ALS clinical trials, you get really lucky. And the drug riluzole, which has been approved for a very long time for ALS, was pure luck that it actually worked. If you talk to Gilbert Bon-Simon, who led the initial trials, that they just happened to end up with that drug and it just happened to work. And now they did a good trial, so that helped. More recently, the Healy platform trial, and I'll come back to this, which is looking in a more efficient way at multiple different drugs, and led by Merit Sokovic and Sabrina Paganoni at MGH, is really, I think, accelerating the development for sporadic ALS. But ultimately, it's hard to do this because there aren't a lot of biomarkers that can be used, and there aren't a lot of ways to validate targets in sporadic ALS. I think that what's most exciting for me has been the drugs lecanemab and donanemab in sporadic Alzheimer's disease, where I think the success really can be attributed to a more precise approach. So there are various different biomarkers, both of amyloid and tau, that have really helped with this development. And similarly, I won't talk about it, but tofersen for SOD1 related ALS is another example where biomarkers really have allowed a more precise approach to clinical development. Well, for tauopathies, really there are three indications that we're thinking about for developing tau-directed therapies, and these are the most advanced in terms of clinical development. And we can think, if we want to do a sort of precise approach, well, first of all, what is the evidence that tau actually causes these diseases? And so, what I want to tell you about, and I'll come back to this again and again in my talk, is that, we have a lot of experience doing clinical trials in Alzheimer's disease, and we have very good biomarkers of tau protein in both soluble and insoluble forms. But there's not a lot of genetic evidence, or the evidence is relatively weak, that tau is involved in Alzheimer's disease. Now, we are very interested in doing clinical trials in human tau mutation carriers, and we know this is a disease that is caused by tau, because if you have a autosomal dominant mutation in the MAPT or tau, human tau gene, you get frontotemporal dementia spectrum disorder. And I'll come back to the challenges of doing trials in this indication. And then PSP Richardson syndrome, or the typical PSP, progressive supranuclear palsy. We've done a number of trials, and we can do good trials in this syndrome. And it's also strongly genetically linked to tau, and we're doing better now with developing biomarkers. So didn't know that Howard was going to be here, but Howard is an author of a very influential paper, which I encourage everyone to read, which is The Rights of Precision Drug Development for Alzheimer's Disease. And Howard, Jeff Cummings, and others really reminded us, looking back at Alzheimer's drug development, that to really be successful in a precision type approach to drug development, you have to have the right target, you have to have the right drug, you have to have the right biomarkers, you have to have the right participants in the clinical trial, and you have to do a good clinical trial. And there are many different ways to go wrong here. Clinical trials are experiments, just like you would do in your laboratory, except for these are much longer, more expensive, and more complicated because they involve humans. And so for Alzheimer's disease, I've bolded some of the things that I think were the key insights that took a while for us to figure out. But the other really rate-limiting step in the drug development of anti-amyloid therapies was an understanding of safety. And we really, I think, didn't understand this very well coming from bench to bedside or into the clinic. And there were these abnormalities called ARIA, amyloid-related imaging abnormalities, which really slowed things down because people, particularly regulators, the FDA, didn't understand whether they were clinically significant or not, and how best to manage that. And that affected multiple aspects of drug development and really slowed things down. And so this is from a recent review where we sort of looked at all of the different amyloid immunotherapies. And again, it was about, depending on when you think they were developed, either in animal models or when they first came into the clinic, it was 25 to 30 years from the first animal experiments to actually the first successful phase 3 clinical trials with lecanemab and donanemab. And one of the key insights in the bottom panel in B is that you need biomarkers not just for patient selection and to measure responses, but also to understand the biology. And one of the key insights was that to really see efficacy or clinical efficacy with these drugs, we had to have drugs and populations where you could remove enough of the insoluble amyloid plaques. And this was measured by a biomarker that was developed at about the same time human trials were starting, amyloid PET. And it was only if you really, really clear out the brain of the amyloid plaques do you really start to see appreciable clinical efficacy as measured by a global endpoint, the Clinical Dementia Rating Scale, sum of boxes. And so it's really these biomarkers that are, I would argue are going to be critical for us to do a more efficient trial. And ideally, we'd like to start the trials, with biomarkers in hand, as opposed to starting the trials and then developing the biomarkers, which one might argue happened with amyloid immunotherapies. So how about tau? Why are we so excited about this as a therapeutic target? And I think many people are familiar with the Braak stages of Alzheimer's disease you see in the upper left-hand panels. And you can see that dark brown stuff in the brain slices of people who passed away is insoluble tau protein. And both the amount and the anatomical location of tau that deposits insoluble tau in Alzheimer's disease is strongly correlated with the severity of disease and the types of symptoms that people had just prior to death. And this is not just a phenomenon in Alzheimer's disease, but also in PSP-Richardson syndrome. This is the color coding here sort of represents different levels of severity related to the amount of insoluble tau, or even chronic traumatic encephalopathy. And so we consider all of these diseases, plus others that I have not shown, as tauopathies, because we think that tau protein may be central to the symptoms and the neurodegeneration, and particularly the accumulation of insoluble tau protein. So where did the idea come from that possibly reducing tau protein might be an effective therapeutic strategy? Well, for me, I learned about this from my colleagues Eric Roberson and Leonard Mookie, but there are others who did similar experiments where they showed that in transgenic mouse models of Alzheimer's disease that expressed a human amyloid precursor protein, that if you genetically knock down the amount of tau protein in these mice, they became resistant to the amyloid toxicity. And so in a mouse with a normal amount of tau, about 15% died after 6 months, but just knocking down or one copy of the endogenous tau actually made the mice resistant to this amyloid toxicity. And they looked at a variety of other neuronal insults, including toxins that cause seizures, and now other people have looked at a variety of different insults and shown that reduction of tau really blocks a variety of different toxins or toxic processes in the brain. And so this is, a potential therapeutic strategy. And now fast forward another, 18 years, and we can see the really exciting data from Biogen and with Ionis, which is up the street from you guys, showing that an antisense oligonucleotide drug, which can suppress tau expression, and here in the lower left, you can see that this is a CSF measure of phosphorylated N-terminal tau protein. So here they're suppressing it at the highest dose for, almost 2 years to about 60% of normal, leads to actually surprisingly in a very, very small number of people. So even a reduction over time in the amount of insoluble tau that accumulates. And so, this seems really promising. We still don't really know clinically whether this will have any sort of benefit. There maybe are some hints and some, very questionable post-hoc analyses that have been done that this might, just the reduction of tau without amyloid might be helpful, but we won't know until an ongoing phase 2 is finished, hopefully in a couple of years, whether the same effect is seen in humans. And whether or not the antisense approach works, there are quite a number of different ways that one might modify tau in a human disease. This is one scheme that we think, how tau causes disease. At first, you have normal tau production. There may be abnormal post-translational modifications. promoting aggregation, and there are endogenous clearance mechanisms like autophagy or the proteasome system, which protect or get rid of these aggregates. But if they're not destroyed, they can transfer possibly transsynaptically to other cells where they can template more aggregates. And again, if they're not cleared, they can lead to toxicity and even larger tangles and large aggregates, which may be, either cause the toxicity or byproducts of the toxicity. So we mentioned that one type of therapeutic approach are the antisense oligonucleotides. There are now more advanced RNAi therapies and gene therapy techniques that probably will be coming into the clinic in the next few years. There have been a variety of different small molecule enzyme inhibitors that are mostly targeted at blocking post-translational modification or aggregation of tau. There are also increasingly drugs that we expect to be in the clinic soon that can actually enhance the clearance through the proteasome system and possibly other mechanisms of tau. And finally, immunotherapies, which are the most advanced, either active vaccines or anti-tau monoclonal antibodies that are thought to potentially block this transcellular or transsynaptic spread of tau. And I'll come back to these because in clinical development, these are really the most advanced and we have the most experience with them. Unfortunately, none of them have worked yet. So to remind you, we said we had to talk about tau genetics. Just for those who are not familiar, the tau is an alternatively spliced gene, and the main exon that we care about is exon 10 because when it's present, we have 4 microtubule binding repeat tau or 4R tau, and when it's absent, we have 3 microtubule binding repeat tau. And in the normal brain, we have a normal adult brain, I should say, we have an equal ratio of 3R to 4R tau isoforms. And similarly, in Alzheimer's disease, even in aggregates, there are similar ratios. But in certain tauopathies, including corticobasal degeneration and progressive supranuclear palsy, there's much more 4 repeat tau than 3 repeat tau. And in contrast, in Pick's disease, another cause of frontotemporal dementia, there's more 3 repeat than 4 repeat tau. And so there are rare families around the world where they have point mutations in the tau gene that lead to autosomal dominant familial early onset dementia. And interestingly, depending on where the mutation is in the gene, it may have a different mechanism and also often will lead to a different clinical phenotype. So for intron 10 mutations, which lead to increased splicing in and increased 4R tau production, interestingly, many of these individuals, when they become symptomatic, have a syndrome that looks like sporadic CVD or PSP. And when you do a genome-wide association study of these sporadic tauopathies, you find that the strongest genetic risk factors are regulatory regions in or around the tau gene. And so this suggests that while, in most cases, tau mutations don't cause CVD or PSP, there are very strong genetic risks. And more recently, it's been shown that there are protective factors that seem to act particularly to clear tau. So TRIM1117 is a ubiquitin ligase involved in tau clearance, and LRRK2 protective SNPs also for PSP might play a role in autophagy. And so to remind you, for most tau drug development, most of the mouse models and many of the iPSC models use P301L or P301S tau mutations, and these cause in humans most commonly a behavioral variant FTD syndrome, but they don't cause Alzheimer's disease. And so again, the links to Alzheimer's disease are a little bit more tenuous with tau. What about tau protein structure? I think most people are also familiar that over the past 5 to 10 years, there's been really amazing technological advances in cryo-electron microscopy, where the 3-dimensional structure of the tau protein aggregates that you can see in a postmortem brain specimen has been solved. And we can see that the core of the aggregate structure is really in the microtubule binding domain and of the tau protein. And or in the C-terminus, and that the N-terminal regions that we measure with the typical Alzheimer's disease biomarkers are not in the key sort of what we think to be toxic part of the molecule. And the really amazing thing, even more recently, is that the three-dimensional structure of this core differs in the different tauopathies. So there's one three-dimensional structure in Alzheimer's disease and a different structure in progressive supranuclear palsy. And whether this plays a role in the different phenotypes or the different cellular vulnerabilities, it is unclear, but it's an important consideration for clinical development. Because the first, as I mentioned, the first anti-tau antibodies that were tested, or these are 4 of them, all targeted the N-terminus, which we now think not to be really associated so much with tau toxicity, and all of them really failed to demonstrate efficacy in human clinical trials, either in Alzheimer's disease and in some cases in PSP. But there are now second generation antibodies that are targeting more of these MTBR and C-terminal regions, and there is hope that because these are thought to be more involved in toxicity, they might be more successful. The problem is that, as you can see, the structure differs in different tauopathies in this region. And so if the epitopes have a predilection for one tau structure or another, and recall that they were developed in tau transgenic mice that are derived from FTD mutations, that it may be necessary to look at various different tauopathies to find the best human disease that responds to a immunologically targeted agent. So what have I told you? That really, what are the three most advanced clinical indications for developing tau therapies are Alzheimer's disease, progressive supranuclear palsy, and tau mutation carriers. And we can rank them in terms of their relationship to tau genetics and to preclinical models of tauopathies, with Alzheimer's disease being the least related, but tau mutation carriers being the most related. So now I want to tell you a bit more about the other aspects that we think about in terms of precision approaches to developing tau therapies. And for Alzheimer's disease, when we think about clinical trials for efficacy studies, they have to be pretty large, on the order of thousands of people, and they take roughly 18 months, but they are very good biomarkers, as many people know. This is a pretty common disease, so it's easy to recruit. PSP, actually, we can do more efficient trials. You need about 10-fold lower numbers of patients. The trials can be faster, but we don't have as good biomarkers. And for tau mutation carriers, these are very, very rare. And so at best, we may be able to recruit a few hundred to do a clinical trial, but we still don't have very good biomarkers. And this is reflected by the number of clinical trials that are in progress for these different indications. So just as an example, again, these are, what a newer version of what people call the Cliff-Jack curves for Alzheimer's disease. So I, really find the exciting thing about doing clinical trials in Alzheimer's disease is that we have biomarkers that you can look at, either soluble forms of amyloid or insoluble forms of amyloid plaques, or soluble tau or insoluble tau, and relate these to the different features of disease progression and disease onset, and particularly for Alzheimer's disease, but likely for other tauopathies, it seems to be really the emergence of substantial levels of insoluble tau in the brain, what my colleagues Risa Sperling and Keith Johnson call the catastrophe, that is related to the onset of clinical symptoms in Alzheimer's disease. And Keith and his lab have shown, using tau PET, that there's really an acceleration of insoluble tau deposition. This is, again, a representation here you see in preclinical Alzheimer's disease. People are just about to become symptomatic. You see an increased amount of tau deposition, and then once people become symptomatic, an even faster rate of insoluble tau deposition. And so the goal for many Alzheimer's trials is to intervene with tau therapies right as early as possible in or before the catastrophe to really hopefully blunt this increase in tau. And what we've seen with, these are data that also I think many people have seen, that you can even use tau PET in many cases and as a potential biomarker endpoint, not for approval, I would say, but to look to see whether a drug is having an effect. And with lecanemab, which is now approved for Alzheimer's, the anti-amyloid drug, you can see that in the phase 3 trial, there were in the tau PET sub-study, there were actually significant attenuation of tau accumulation in medial temporal and temporal lobe regions from amyloid removal early enough in the disease. And so this suggests that this is a useful biomarker. And again, another example of why biomarkers are really useful and exciting in Alzheimer's disease is now, again, as most people know, there are blood tests. We can measure plasma p-tau species 181, 270, 217, many others now, and that with both lecanemab and with donanemab, you see very prominent reductions and relatively rapidly in response to amyloid removal. And I won't go into what people think about the mechanism is, but this suggests that you really can take this precision approach for Alzheimer's therapeutics. And so, if we try to guess what's going on or to demonstrate with biomarkers how the new anti-amyloid therapies worked in the phase 3 trials that were positive, well, we intervened relatively late after symptom onset with the anti-amyloid antibody. We saw very, very prominent reductions in amyloid plaques. And very sort of modest reductions maybe in tau PET in some people, and a modest effect on cognitive decline. But, I think that one could argue this might have been predicted by the biomarker changes if we had had those biomarkers much earlier in the course of drug development. So now I want to switch to progressive supranuclear palsy, the next tauopathy. Since this is an Alzheimer's center, I'm not sure how many people have seen these patients. So I thought I would show a quick video. This is of some of the original patients that were characterized by J.C. Richardson, who was the neurologist on the team. John Steele, actually, who is the first author of this paper, just passed away a couple years ago and shared a lot of his slides in this video with me. So here's a video.
In the 1950s,
this is John Steele narrating.
Neurologist Clifford Richardson identified an unusual disease whose hallmark was progressive supranuclear gaze palsy. His first patient, a Canadian executive, could follow to either side through a full range. This film was made in 1959 when the patient was 56 years of age. At this time, the face was spastic. He was dysarthric. He could not look up and down, and he was quite unable to follow Dr. Richardson's pen, moving slowly in the vertical plane. But when his head was passively flexed and extended, his eyes moved through a full range reflexly in the opposite direction as occurs when a doll's head is similarly positioned. Here he is attempting to look down at his watch, but as you see, he is unable to do so. When his neck is flexed, however, his eyes deviate up and he cannot move them down. Richardson's second case.
So I think in the interest of time, I'm going to just keep going, but lots of exciting videos to see, and these are available online. So John really spent his whole career thinking about PSP, and it's a very characteristic, the Richardson syndrome, which we named again for J.C. Richardson because he was the first one to really describe it. And that first patient was actually one of his friends who was originally thought to have Parkinson's disease, but he was very astute and said this isn't Parkinson's disease. But anyway, it's such a distinctive syndrome that even the non-neurologist Charles Dickens could recognize it. And this is a description in one of Dickens' novels of a man really, I won't read it to you again in the interest of time, but who seems to have, this is sort of the perfect description of Richardson syndrome. And it turns out, actually, if people are interested, John Hardy told me this pub still exists. off the northern coast of England, and people can go there and imagine as someone with PSP coming in and having a pint. One of the things that frustrates me is that even though PSP is a rare disease, it's about as prevalent as ALS, but it's a really, I think, understudied disease, and we could discuss later why I think that is. But the prevalence of both PSP and ALS are about 8 in 100,000 if you look at different meta-analyses of epi studies. But there's a lot more work. These are the number of epidemiological studies, about roughly 10 times more in ALS than in PSP. And so we're trying to change that because this is really an equally terrible and fatal disease. We have good clinical rating scales now for PSP. This is one of my patients who had path-confirmed PSP. This is the PSP rating scale. Again, in the interest of time, I won't show you all the videos, but she was a dear, dear woman nurse who volunteered to be the first person to receive a tau antibody for PSP in a clinical trial.
Started out in the early stages of the disease, being able to sleep anytime and all the time. And then I got where, for whatever reason,
so you can hear there's a very characteristic dysarthria. She also starts to cry a little bit later. And she's talking about her difficulty with sleep, and there's a very, very characteristic sleep deficit.
Quite often.
About the trouble that you have with.
So, what about clinical trials for PSP? Well, on the left, this is a trial. This was actually a BACE inhibitor that was done by Merck, and you can see that in this 18-month clinical trial, the number of patients per group to test the clinical efficacy of the drug in mild to moderate Alzheimer's disease was about 620 to 30 per group. And in this trial, according to the paper, they had 90% power to detect a 35% treatment effect on slowing clinical decline. In PSP, we need roughly a quarter of the number of patients, and we only need to follow them for about a year to have similar power. You can see that in a trial of a drug that didn't work, called terbinafine, we had similar power, but the trial was much smaller and faster. And so one advantage of PSP, even though we don't have the biomarkers of Alzheimer's disease, is we can do a more efficient clinical trial. And over the years, we do have some biomarkers. You can see that there are very prominent rates of brain atrophy, particularly in the brainstem of PSP. And this has been replicated many times. It is now used in clinical trials. So the midbrain volume decreases significantly over the course of a year. And now we can expand our studies to the disease corticobasal degeneration, because we can apply those same blood tests for Alzheimer's disease to help to parse cases of corticobasal syndrome, which is sometimes caused by Alzheimer's disease, into cases that are truly caused by likely CBD pathology or atypical Alzheimer's disease, using plasma biomarkers, which produce similar responses to amyloid PET. And when we do this, we can also look at volumetric tools to give us greater confidence that someone truly has a CBD pathology. This is sort of combining midbrain atrophy measures with cortical atrophy, and particularly CBD patients have a lot of cortical atrophy as well. Even more exciting now that we know that there are these different structures of tau aggregates in the different tauopathies. Chihiro Sato and Kanta Hori, working with Randy Bateman, have actually developed specific biomarkers that or antibodies that can bind to the altered three-dimensional tau structure in CBD and PSP. And while they're not quite ready for prime time yet, you can see that either in brain tissue, you have really selective binding of these some of these antibodies to CBD or PSP tau as compared to Alzheimer's disease tau. And interestingly, some of the tau mutation carriers also have similar binding, and likely they have a similar structure. And you can start to develop these into CSF biomarkers. And I'm not sure that they're quite ready yet, but hopefully soon, at least for CBD. And so using these new biomarkers, we can also extend some of the work from PSP into hopefully corticobasal degeneration trials. And I know there are others. Irene Litvin at UCSD is very interested in this as well. And so we think we can start to use new tau biomarkers to do more precision trials in different non-Alzheimer's disease tauopathies like corticobasal degeneration. So maybe you could screen with a plasma blood test to rule out Alzheimer's disease, refine the population for inclusion in a trial with volumetric MRI, and then really to confirm that someone truly has CBD or PSP using a novel CSF biomarker. So now I want to mention a bit about tau mutation carriers, and obviously these are the individuals who are closest to tau transgenic mice because they have some of the same mutations often that the preclinical models have. But the challenge here is that these families are very, very rare. And recently, we've actually updated this survey. I've done a global survey of different tau mutation carriers, and probably globally now there are fewer than 1,000 carriers who might be eligible for a trial. And once you screen them, we think, many fewer. So the real challenge here is recruiting enough individuals to do a clinical efficacy study. And we have to think about more efficient ways to do this for tau mutation carriers. But that's, work that's really underway now. The other challenge with tau mutation carriers, as I mentioned before, is where the tau mutation is in the tau protein itself can affect the clinical phenotype. And you can see, for instance, that certain tau mutations that lead to more 3R, 4R tau, like Alzheimer's disease, tend to have a more Alzheimer's disease-like clinical picture. And even tau PET ligands like tau fluorotaucipir bind to tau in these individuals, but not in people with like P30L or the most common of the FTD-causing tau mutations. And these people also tend to have slower rates of disease progression. All this being said, we can apply Bayesian disease progression models, and we can now start to look at all of the aggregate data that we've collected longitudinally in tau mutations in various networks, both in North America through the NIH-supported All-FTD network, and in Europe and Canada through the GenFi network. And we are starting to develop basically the same sort of Cliff Jack-like disease biomarker disease progression maps. And we can see that in tau mutation carriers in aggregate, that we can measure changes in neurofilament light chain in blood and MRI years before the onset of symptoms. And so we are continuing to refine these tools and to develop new biomarkers for this symptomatic phase, where we think it really will be necessary to develop very well-powered trials in these rare individuals to test tau therapies. And so, for instance, one thing that we're doing, this is actually progranulin mutation carriers, is looking at plasma neurofilament and other plasma biomarkers more frequently. In the data here, these are based on annual clinical and biomarker collections, but we are now starting to collect things every 3 months through something called the Neurofilament Surveillance Project, which is a public-private partnership funded by a number of foundations and industry that is operating in conjunction with All-FTD. And we think that these more frequent biomarker measurements might allow us to get more precise tools, although you can see that even when we look at more detailed in time maps of longitudinal change in neurofilament in plasma in progranulin mutation carriers, this is not tau, that it's pretty noisy, and we probably need better plasma biomarkers. We can use plasma biomarkers also to screen people in early onset populations to try and understand the causes and identify people from diverse populations who might be eligible for our trials. We have a project funded by the NIA called Beyond Biomarker Evaluation in Young Onset Dementia from diverse populations, which is actually measuring blood biomarkers of neurodegeneration in individuals' homes, along with digital biomarkers. And this is now actively underway. We have a very active social media and web-based presence for recruitment, as well as a lot of different community-based efforts for recruitment. These are some of the landing pages you can see. And what happens is that people actually are recruited through different community-based projects. They register for the project online, and eventually we do an at-home blood draw for different blood tests and a tablet-based brain health assessment administered by the phlebotomist, and then they are referred and invited to come into sites for further, most of them are ADRCs for further evaluation. This project really didn't get started till this year. It's gone amazingly well to the point that we probably are having to go on hold for recruitment. We recruited about over 200 people in the first couple months, and even in the first 20 people who we actually have complete data sets, we found 2 people, one who likely has early onset Alzheimer's disease. She's 57, a very elevated p-tau217, normal neurofilament, abnormal digital assessments, and another individual who's a gentleman, also about the same age, with very high neurofilament, normal A-beta, p-tau, and there are some concerns this person could have frontotemporal dementia or ALS. So in the last few minutes, I just want to tell you about more efficient approaches to clinical trials. Many of these ideas come from oncology. One idea is that instead of doing multiple successive clinical trials in these different diseases, one more efficient way is to put all of these different tauopathies into one clinical trial or one basket, and then use possibly a tau-related biomarker to look for the efficacy across multiple different tauopathies of a particular tau-targeted drug. And I'll show you an example of this. And more recently, this is the idea of an umbrella trial or a platform trial where we're testing multiple different drugs or multiple different therapeutic approaches in the same tauopathy. And I'll tell you about 2 new projects that are going on for this. So this is an example of a basket trial that we did a number of years ago, where we combined patients with Alzheimer's disease, corticobasal syndrome, and PSP in the same phase 1 program of a microtubule binding agent derived from a taxol called abiotaxane. And here you can see that at baseline, we could demonstrate, we could use biomarkers and either both the brain atrophy as well as CSF biomarkers to show that each of these populations actually likely did have the disease of interest. And although the drug didn't work for any of these 3 populations, we found that the safety profile was really quite different. And then interestingly, the Alzheimer's patients had very severe immune reactions, including cases of anaphylaxis that we did not see in either CBS or PSP patients. So that's an example of the power of a basket approach. The other idea is a platform trial where instead of, in traditional drug development, we think we do a clinical trial where we have one placebo group and one treated group. And, you can think that, well, for instance, in a hypothetical trial where we had 45 in each group, and it took about a year and a half to do each trial, it would take us about 450 participants followed for 13 years to test 5 treatments. But what if we tested 3 treatments at the same time and we shared this placebo group across all 3 treatments, and then we could really do this with fewer participants and in half the amount of time. And so that's the potential power of a platform trial or umbrella trial approach. So we're very pleased to say that we are now funded by the NIH to do a platform trial in sporadic Alzheimer's disease. There's already a platform trial in genetic Alzheimer's disease that's called DIAN-TU. But the idea with the Alzheimer's tau platform is that we're going to use these same biomarkers. This is a phase 2 biomarker proof of concept study to really interrogate the amyloid tau neurodegeneration hypothesis, or ATN, of Alzheimer's disease. And instead of just intervening with an anti-amyloid drug, we're going to also introduce different tau therapies. And the idea is that by combining the anti-tau with the anti-amyloid therapy, we might see a larger effect on tau PET and hopefully a much larger effect on cognitive decline. So that's the hypothesis, and the trial is scheduled to begin next year. So this is the initial design. What you can see is that the plan is to have placebo, although this may or may not end up in the final design, an anti-amyloid therapy alone, anti-tau therapy alone, combination therapy, a second anti-tau therapy, and a second combination arm. And then we'll look for the various effects and try to determine which is best. Is it possible that you don't even need to remove amyloid, and then if you have the right anti-tau therapy, that might be sufficient? And so we're really excited to begin this next year in the Alzheimer's Clinical Trial Consortium. We also are planning, hopefully next year or early in 2026, we expect this trial to start hopefully with a little bit of a lag, but this is going to be in progressive supranuclear palsy. And this is a more traditional platform trial similar to the Healy platform for ALS, where we will look at 3 different treatments for PSP, either anti-tau or other approaches in parallel. And each individual will first be randomized to a treatment regimen, so one of the 3 treatments, and then within that regimen, randomized 3-to-1 drug to placebo, and we'll eventually pool the placebos so that you can compare placebo, equal amounts of placebo with treatment in each arm. So we're really excited about this one as well, which will be funded by the NIH. So just to conclude, what I told you is that unfortunately, or maybe fortunately, depending on how you think about it, tau is a really important protein for neurodegeneration. And what we're trying to do is to develop tau therapies in a much more efficient way than what was done for amyloid immunotherapies. And we can learn that really, we think a precision approach is going to be the best way to do this. And we want to think about precision both in terms of the disease we want to treat, but also the biomarker approaches that we can apply, as well as the different clinical trial designs, some of which being hopefully a lot more efficient than just testing one drug at a time against one placebo. And really what I think and what our goal is to have a basket trial that would include multiple umbrella trials. So we would like to have multiple drugs being tested at the same time for PSP, Alzheimer's disease, and eventually tau mutation carriers. And we think by doing this, tau universe of clinical trials, that will really find effective treatments for all of these diseases, or at least that's our dream. So thank you very much for your attention. Many people, both at UCSF and elsewhere, have helped with this work. These are some of the key collaborators. Want to thank the NIH and many foundations for support of this work. Also particularly want to acknowledge my colleagues at the Alzheimer's Clinical Trial Consortium and other individuals, including Irene Litvin at UCSD, who's been involved with the different platform trials that we are about to begin. And I think we have a little bit of time for questions, so I will stop there.